Okay, I usually try to put up a little synopsis of what we're doing. I'm going to start out with just backing up a little bit and just showing you a typical example of evolution, evolution of a horse, which I'm a horse lover. So, like, evolution of the horse, the typical ones that is followed and talked about. Good morning. So, let's just do this, and we won't spend much time. Then we're going to go talk about the origin of life, which is going to be very simplistic. I'm not going to do a whole chapter. And then we're going to go back to the main chapter for today, which Aaron, very nice to give us a really detailed, Aaron Ellis, a very detailed chapter 26 about phylogeny. So, evolution of a horse. Got a lot of light coming in somewhere. Can you see that at all? Yes. We do still have some lights on, I guess. Anyway, in terms of the evolution of the horse, about 55 million years ago, and you usually see it abbreviated as MYA, or sometimes you'll see BYA, and now which is millions of years ago. About 55 million years ago, there was a very small, the original ancestor of the horse. It was this particular animal that was multi-toed, 4 or 5 toed, small animal. And you can see from 55 million up to about 45 million, so 10 million years, then we got into something called a Mesohippus. It still wasn't the genus and species of the horse that we have today. And then you see about 35 to 30 million years ago, we ended up with this one. And the number of toes kept reducing. And the size of the horse kept getting bigger. And here is the most recent transitional form at about 20, 25 million years ago. You don't need to know the names of these. Okay, and now today we have Equus for equine, EQUS, which is the genus name for the horse, and we have it looking, it has one hoof. Now you still have the remains of a couple toes, the fetlock joint, but it's really, they're one-hoofed, one-toed ungulates, which is what they're called in terms of groupings of mammals. And in terms of variation, I just wanted to show you, wow. I don't know if you can see, it kind of looks like a bulldog.
That's a halter horse.
Bulldog. So what breed is that? That's a quarter horse. A breed is a sort of almost like a subspecies.
That looks more like a quarter pony.
Same with dogs, right?
I'm sorry, what?
Same with dogs.
Yes, same with dogs.
That'd be short boy.
You know, you have your German Shepherds, you have your Collies, you have, these are just different breeds, but they're all the same genus species. It's just that this is a quarter horse, a very, very, very heavily muscular quarter horse.
That's
probably
boy.
is used at least for halter conformation. Yeah. This is an Arabian horse. Now the colors, a lot of different breeds can have a lot of different colors, so don't make the color as a signature of a particular breed. But see that, how the shape of the head is smaller, the end of the nose, the nostrils usually more flaring. It's a shorter back, it's a more lean type of horse, very showy. You know, the tail comes up. Arabians usually have a lot of spirit. is actually a thoroughbred, which is a bigger horse. They're measured in hands. Anybody know how many inches a hand is?
4.
4, that's right. So this one could be, what, 16 to 17 hands easily, which that'd be 16 times 4. That would tell you how many inches from the ground to sort of the top of the shoulders, which is called the withers. Big horses. Notice that that is more like a quarter horse. In fact, quarter horses were originally through artificial selection. I think between thoroughbreds and breeding with others in order to actually create the original quarter horse. Here's a standard bred. Now, this could be used for, like, the Kentucky Derby runs thoroughbreds where jockeys are on their backs. But you can also use them for hunting, jumping, dressage. It's a long way to fall. And standard bred is along the lines of the thoroughbred, but not usually quite as tall. There are different conformational differences, because you have those judges that know, just like with the dog shows. You can judge them on the basis of conformation, whether or not they are, how close they are to the standard conformation. The standard bred pacer, this is a pacer, which means it does a pacing, and it's used for harness racing, although there are trotters, and if you go to the harness races, you'll know there are pacer type of races, and there are trot. The trot's sort of a diagonal, moves forward and back at the same time, whereas a pacer, they move on the same side. These two go like this. A pacing, if you're riding it, would be a lot smoother than most trots. And then you see an Appaloosa, which can have all kinds of different patterns. This is one that has a blanket with white, of course. A lot of we used to see a lot of those associated with Native American horses, but also the paints and pintos as well. This is American paint. Paints and pintos have, and then there are different variations of those designs as well. But this is a different breed. Did you know within an hour or two, when a foal is born, it will get up and walk, which is a lot different than a baby human. And it's really very neat to watch, because they're as wobbly as can be. But they do stand, and they're expected to soon after birth. And there's a Morgan horse, which is one of the original, was made in the USA, Morgan horse breed. It's Justin Morgan, who is supposed to be the artificial selection breeder for this particular, it's a little smaller horse. Ponies are usually less than 56 inches to the withers, and horses are usually above that. This is just showing you some of the different colors. I know you can't see it real well. There's a whole array of colors. And then this is just a page I found on horse genetics about the different colors and how they're created. You don't need to know the genetics, but I'm just telling you it gets complicated. It is a poly. Is really important, because I had a sorrel horse. Pretty sort of reddish brown. She had a really light tail. hoping to get a Palomino, which you can, there's like a 50% chance, and I ended up getting another sorrel, which was fine, but. So, definitely artificial selection, you talk about breeding, you're selecting from certain traits, like I was trying to get a Palomino that way, but, sorrel is, I love it too.
I have horses. I have horses.
You do now, or you used to?
I did.
You did. How many of you have had horses, or you've at least worked with horses? Okay, very good. They've always been my first love. You know, I was raised on a farm in southeastern Ohio, and with about 165 acres. Southeastern Ohio is very, very rural, very agricultural. Down, where is that? Morgan County, which is one of the 88 counties. It's south of Zanesville, east of Athens, and north of Marietta. So it's sort of, but 4-H, being a member of 4-H was the highlight of my childhood. Looking forward to the county fair every year and showing. I also showed cows for a little bit. Jerseys, normal cows. But we also had sheep and hogs on the farm. But unfortunately, my dad got some health issues, so we moved from the farm when I was 12, 13. But I still had horses for quite some time after that. Okay. Would love to have one now, but I don't think it'll fit in my backyard. Thank you, Brent. Okay, so there's nothing. What should you know about the horse? I would say it's been over 50 million years of developing to the species of horse we have today. It went from like a 4 or 5 hooked toed mammal to a 1-hoofed mammal, and it got much bigger. Those are some of the things that I would remember about that, but I thought it was kind of fun to show that. Okay, we're supposed to at least address the origin of life. And there's a whole chapter, and I'm not going to go through the whole chapter. This is a page taken out of an advanced placement biology prep book, and I just wrote my own notes, kind of in it, to give you a good idea. So starting here, how life probably emerged. Now, we're talking, evolution is a theory which, to a biologist, is a broad pattern that has been substantiated by all the lines of evidence time after time after time. So it's a theory, which is the biggest deal you can make, without, going into narrow-mindedness and saying this is the truth. We don't use the word truth. But if you were to use the word truth, the theory is, okay, as close to it as possible. This, I would say, on terms of the origin of life, is really hypothetical. So if you're really kind of thinking out loud in regular conversations, good morning. Really, a lot of times when we say, well, I'm theorizing that, really most of the time we should be saying hypothesizing. I get so mad at watching TV, because these people are talking about, well, I theorized that. But to a scientist, it drives us nuts because you're not theorizing, you're hypothesizing, because you don't have that much evidence, you're just talking. Okay. Anyway, this is a model that has been hypothesized. Steps of the heterotroph theory. So if I were to label this, would be the origin of life, the first form of life was probably a heterotroph. What do heterotrophs eat? Troph means feeding or whatever, and hetero means other different, so it's an other feeder. So are we heterotrophs or autotrophs? We're heterotrophs because we eat food, we eat other things. The main producers or autotrophs are the plants. Here we go, we're starting ecology already here. Okay, are the plants, and the plants by photosynthesis make the original source of food and the original oxygen. Well, this is a possibility. It's our the Earth was formed, we believe, based upon lots of different evidence. Approximately 4.5 billion years. And it was a very violent, turbulent time on Earth 4.5 billion years ago, where most of, you didn't have anything called life. There was just a lot of physical heat, volcanoes, all kinds of situations. Now, volcanoes release gases to the atmosphere, and we believe that the gases that were released in the atmosphere were ammonia, methane, water, hydrogen gas, diatomic hydrogen gas, carbon monoxide, and carbon dioxide. What is missing from there that we need? Oxygen. So the big thing to know is there was no oxygen. And that makes sense because if you have oxygen in the atmosphere, you can have oxidation, sort of like rustling, sort of like corroding, sort of like changing. So if there are macromolecules, which would be like carbohydrates, or even the monomers of carbohydrates, lipids, proteins, and nucleic acid, if those monomers could form somehow, they would have broken down probably if there would have been oxygen in the atmosphere. Okay. Gases condense to form CO2. So you had water. You had the extreme UV rays from the sun and lightning, constant lightning. So sparking with heat, with volcanoes. And actually, we believe that there were simple organic molecules that were actually formed, like amino acids, the monomers, the fatty acids, the glycerols. autotrophic, whereas fungi are heterotrophic. Okay, so those are the first ones we believe. Well, because of all this going on, you know that a lactic acid, not lactic acid, in alcoholic fermentation, there's a second, in alcoholic fermentation, besides the production of alcohol, what gas is given off? Carbon dioxide. Okay, so carbon dioxide, the buildup of carbon dioxide from anaerobic respiration sets up photosynthesis, because what is the main gas that photosynthesis uses? Carbon dioxide. Okay, question, Brooklyn.
Yeah, I'm just confused on how are heterotrophic prokaryotes the first forms of life when we said that protobiomes were the first living?
Okay, no, those are sort of like the intermediate forms. They're not quite alive yet. They're right before life. It's sort of like saying something's cancerous or it's precancerous. So the protobiont condition is sort of like pre-living. It's like the transitional step right before actually making cells and living things. So good question. I'm sure other people had the same. Okay, then there were some of these prokaryotes that probably mutated with some mutations and evolution. Of course, prokaryotes are bacteria, right? They don't have a true nucleus. They don't have membrane-bound organelles. But there were some prokaryotes that became autotrophic, which meant, okay, we got the CO2, and I just happened to create a way to use the CO2. Okay, so autotrophic prokaryotes appeared when mutant heterotroph gained ability to produce its own food through, I think, the most magnificent process that exists in biology that we owe our lives to is photosynthesis. Because without photosynthesis, we don't have any food, and without photosynthesis, we don't have any oxygen. So if something happens on the Earth that the sun turns off, of course we'll freeze to death too, but if the sun turns off or something happens and plants are killed by hazardous waste in the environment, toxic environmental conditions, or whatever, in fact the most oxygen is produced in the oceans where the phytoplankton exists. So the more we keep dumping, I'm sorry, I'm going to say crap, whatever, in the ocean, the more phytoplankton we kill, the less oxygen that we're going to have being produced, and that's going to affect the food chain. Oh, I can't stay away from ecology, can I? Okay, so you see how evolution and ecology just kind of, whew. So that's why we're going to ecology pretty soon, next week. Okay, so then, because of photosynthesis, the release of oxygen in light reactions, the photoreactions, remember? The splitting of water, punch water in the belly, hydrogen gets accepted by NADP, and oxygen goes out through the pores in the leaves and goes into the atmosphere in the light reaction. So the oxygen in the ozone layer appeared, and ozone is like O3 instead of O2, which is diatomic oxygen. And the ozone layer helps protect us. Another environmental thing we're concerned with is the degradation of the ozone layer, because it helps protect us from some of those harmful ultraviolet rays that are coming in. So ozone layer actually helps to support and protect life as we know it. And then there was a researcher by the name of Lynn Margulis who said there were symbiotic relationships that existed between these prokaryotes, and some inserted themselves into other cells, and we actually had the evolution of eukaryotes. The eukaryotic cells, EU, which means true cells. True nut, we're really true nuts. The nut is the nucleus. Okay. So,
symbiotic relationships of prokaryotes led to the creation of eukaryotes, and that theory is called, and it's a theory, the endosymbiont theory. And did you learn in the first semester what's special about mitochondria and chloroplasts?
They were originally prokaryotic organisms of their own that evolved into being part of eukaryotic cells.
Exactly. And what do they have? DNA. They have a circular DNA, which is what prokaryotes have for their main chromosome. They have ribosomes also. They're not exactly as sophisticated as the ribosomes in eukaryotic cells, but for protein synthesis. So mitochondria and chloroplasts actually have their own DNA, and in fact, in some of the DNA ancestry type of tests, they sometimes, some will look at mitochondrial DNA, and that can help trace the original ancestry even a lot better. Yes.
Isn't mitochondrial DNA usually matrilineal?
It's usually what?
Mitochondrial DNA are usually matrilineal.
Yes. From the mother?
Yeah.
Yes. Yes. It's from the female side because during mitosis, the cytokinesis, before it occurs, all of the organelles are doubled. And so that when the cell splits into two cells, it tends to have more of the maternal lineage. But when we talk about the cell reproducing, we're talking about obviously the nucleus. It would be in a eukaryotic cell, the DNA in the nucleus. Okay, and then look at this. Look, 2 words. Oh, our favorite subject.
Selection.
Natural selection. And life continues to evolve today. And we believe in biogenesis now, which is life comes from other life. However, I kind of said up here, this is an abiotic. So abiogenesis was possible back here because we didn't have oxygen to break stuff down in the atmosphere. Whereas today, abiogenesis, which is the same thing as spontaneous generation, should not be possible. Okay, so you're saying, okay, that's a lot of stuff. Please turn to the next page. The origin of life. This is the written summary, and it tells you the people. And the first person that you should know, I'm sorry, I forgot to show you the word for security reasons. So anybody, if I forget, just feel free to get up and check it. Operin was a Russian, I say these sometimes, I want you to know that research comes from the whole world. Operin, O-P-A-R-I-N, was the person back in 1920 who originally hypothesized that of what the atmospheric gases were and how life could have existed. Well, he didn't really research it. That was his hypothesis. Then the next guy I would remember is Stanley Miller, who was at the University of Chicago as a graduate student. And in the 1950s, he tested Operin's theory, or hypothesis, and proved that almost any energy source would have converted the molecules in the early atmosphere into a variety of organic molecules, including amino acids, because he actually collected them. They used electricity to mimic lightning and UV light that must have been present in great amounts in the early atmosphere. Okay, the next person is Sidney Fox, and we're recent years, he carried out similar experiments. However, remember, like we said, things were never done because with new technology and so forth, we continue to add to our theories. Sidney Fox, he began with organic molecules and was able to produce membrane-bound cell-like structures that he called protenoid microspheres that would last for several hours before they run out. Protenoid microspheres. And then the bottom just goes ahead and puts in words what we pretty much already said. I brought a lot of what was here and put it on the front for you. There are several points to prove, or let's not say prove, but support. If you're a true scientist, you don't like to use the word prove. It supports that mitochondria and chloroplasts are endosymbionts. Chloroplasts and mitochondria have their own DNA. We already talked about that. DNA is more like prokaryotic DNA than eukaryotic DNA. It is not wrapped with histones. Histones are the proteins that human and other eukaryotic chromosomes are wrapped with. They're like spools, like thread on spools, and those proteins are called histones. So prokaryotes don't have those. These organelles have double membranes. Why? The inner one belongs to the symbiont. Okay, the bacteria that was mitochondria bacteria or chloroplast bacteria. The outer one belongs to the host plasma membrane. The theory states that the chloroplast and mitochondria were taken out by the host cell by some sort of endosymbiosis process, such as, remember in the cell chapter and the transport chapter of semester 1, you learned about different types of endocytosis, and one was an engulfing line, surround the plasma membrane and go and bring it in, and that's called what? Phagocytosis. It's also what a white blood cell does to a lot of bacteria and invaders. Phagocytosis is important. Anybody remember what it's called when? See, I like to keep writing stuff back. What's it called if it's a liquid? What is it? Pinocytosis or pino. I usually say pino. You're more right with the way you're saying it. But I usually say pinocytosis because it rhymes with, I think this is a word for it. It rhymes with wine and wino drink. Pino, wino. So if you try to figure out which one, what do you call it? Pino or Pinot is drinking liquids, and phagocytosis is eating or engulfing food. All right, so this is a very detailed chapter, and I really commend you on digging the very important parts out, and you've done very well with that. But you have to also understand that the figures, the illustrations in the book are also very important to go with this. So let's just look at the basics of this. Phylogeny and the tree of life. Investigating the tree of life. We already know what taxonomy is, right? Biologists classifying kingdom, domain, kingdom, phylum, class, order, family, genus, species. Relationships between life and its diversity examines patterns and products of evolution. Patterns and products. What is phylogeny? It's the evolutionary history of a species or group of species. So definitely highlight or whatever phylogeny. That's kind of what the title is too. So taxonomy and phylogeny kind of address similar things, but phylogeny tells us more about the evolutionary history, whereas taxonomists have just kind of looked at morphological characteristics and said, well, this one has jaw, this one has a body mostly covered with hair, it has mammary glands, it has whatever, so it must be a mammal. Comparing morphology of how it looks is more taxonomy, but now we're adding phylogeny to the taxonomy, and we've actually changed some things. I don't know, this is probably not going to say it, but I remember when, let's just say when I was in high school, and there were 2 kingdoms of classification, animal and plant. Then there were 3, animal, plant, protista. Then we went through a whole bunch of stuff, and then they said animal, plant, protista, Monera. Monera for the bacteria, because they didn't know what to do with bacteria. And then finally, out of all those kingdoms, the Protus kingdom is the catch-all kingdom. It's the kingdom you put something in because it doesn't really totally match anywhere else. Okay, so you've got amoebas and paramecia. Put them in the Protus. They're animalcules, they used to call them. They're like animals, they move, they do all these things, but they're unicellular. So, and then the algae, they said, well, what do we do with the algae? Well, we used to put them with the plants. Well, most of them are microscopic, or many of them are microscopic, let's say it that way. And so let's put them with the Protus also. So, okay, we'll do that. So we'll put the algae and we'll put the protozoans, little animals, the protozoans and the protista, and then we found slime molds. Okay, they're sort of like somewhere between the protozoans and a fungus. Okay, so there's a group called slime molds. So those 3 groupings are now in protista. And then we said, we used to have the fungi with plants. That's really wrong, because they're heterotrophic. That's a big deal. So we created another kingdom for fungi. Okay, and then since we've done more DNA analysis, we've come up with domains. So Carlos Linnaeus told us the kingdom, phylum, class, order, family, genus, species. But now we have domains. So there are 3 domains: bacteria, archaea, which are bacteria-like, but they're more complex and they usually live in extreme environments. And then all the eukaryotic cell types. So in the eukaryotic cells, the organisms with eukaryotic cells would be in the kingdom Protista, the kingdom Fungi, the kingdom Plantae, and the kingdom Animalia. So there's 4 kingdoms in the domain Eukarya. Has a typical nucleus. The cell has typical nucleus, DNA in the nucleus, nuclear membrane, and membrane-bound organelles. Okay, concept number 1. Okay, there's the taxonomy. This stuff you should already know. This is what we just talked about right here. Now, each of those groupings, a phylum, a kingdom, a genus, a species, are a taxon. They're a grouping, nested grouping. taxa for plural. Greater similarity in taxa indicate a closer relationship between the organisms. So this is pretty much Linnaean. Linking classification and phylogeny. I just threw this down here because this phylogeny, we're talking about phylogeny. Ontogeny recapitulates phylogeny. This is a very, very old theory in comparative anatomy. That development of the individual tends to retrace the steps of the group. Yes. I'm sorry, what?
What does it say after family and genus?
Oh, family, order, family, genus, species. And you know, you can have sub and super, like so you can have super classes, which means a half step. Sort of like the black keys on the piano, it's like a half step, either above or below.
Flat.
Flat, yeah. Sharps and flats. Okay, so if we go over here, phylogenetic trees, a diagram displaying the evolutionary history of organisms. Please read through this, and there will be branch points which represent divergence of two evolutionary lineages from a common ancestor. Does this sound like descent with modification? Okay, how to read a taxonomic tree. Well, there's a figure in 26.5 that would be probably good for you to look at some point. Groups of organisms sharing an immediate common ancestor or closest relatives. There are some other words. Rooted. It's where the branch point furthest to the left represents the most recent common ancestor of all taxa in the tree. There's a basal taxon, which is at the bottom, originates near the rooted common ancestry of the tree. And there's some other words here. But what we can and cannot learn from a phylogenetic tree, trees show patterns of descent, not phenotypic similarity, evolutionary history. So phylogenetic tree is really the evolutionary history. Now, are these necessarily, are they more of a hypothesis or a theory? Hypothesis, yes. And although there's a lot of backing to most of these trees, we still are going to discover more information. What was it? Something I just read yesterday that I thought this could really apply to this class. Or Monday. It shouldn't be assumed that a taxon on a tree evolved from the one next to it. You could read through all of this. It worked. Okay, let's go through a few of these real fast. Like right here. This is a snake, right? No, it's a legless lizard. Okay, now, here is a phylogeny, a tree. Here's the ancestral lizard with limbs. Then you have geckos, and then you have iguanas, and you have monster lizards. And down here, there's one that goes off, the glass lizards, which have no limbs. And the snakes have no limbs. Okay. So you have these branching points. And we know about Linnaeus, we know, I'm going through this fast because I know you know this stuff, right? 3 domains: domain bacteria, domain eukarya, domain archaea. Are you able to do the animations? When you open your PowerPoints, are you able to do the animations?
I don't think so.
Okay, what I found, I couldn't do them either on my computer, and it said, there was a little note down below, it says you need QuickTime, which is an Apple type product. So all I did was download QuickTime, and then it played them. So you might want to go through and play some of these
In
classifications.
the early days of biology,
Oh.
organisms were classified as either plants
Okay,
or animals.
I think we need to go somewhere else. Okay, okay, just, okay, so here you've got an example. Now, this does not necessarily indicate time of descendancy, so there are some misleading things, so don't think that this one happened before this one or this one before this one. It's just, okay, represents a hypothesis about evolutionary relationships. Each branch point represents the divergence of evolutionary lineages from a common ancestor. Sister taxa are groups that share an immediate common ancestor that is not shared by any other group. So, some terminology. And this is one to look at. And these are, of course, we're both primates, the chimps and the humans, and these are sister taxa right here. These are off by themselves. The amphibians are off by themselves, and the fish are off by themselves. They can be drawn horizontally, vertically, or diagonally without changing the relationships between groups, so you can see them staggered. I will try to put at least one phylogenetic tree on the test and ask some questions about it. It's nothing to memorize, it's just you have to learn how to interpret them. Should not be assumed that a taxon evolved from the taxon next to it. That's kind of what I was trying to tell you. And also in time as well. I'm going to skip that one. That one's pretty complicated. I'm not going to worry about that particular one. Phenotypic and genotypic similarities due to shared ancestry are called homologies. Remember, homologous traits come from similar origins because of their ancestry. Like the forelimbs of all the vertebrates. An analogy is usually due to convergent evolution. They're coming from different directions, and because of similar environments, some of their traits may converge and be used for similar functions. That's convergent evolution, and that's called analogous structures, like the flippers of a whale and the fins of a fish. I'm not going to ask you a lot of great detail about how some of this happens. If we had 2 weeks for this one chapter, yes, but we don't. You should know what a clade is. It's a group of species that includes an ancestral species and all its descendants. And cladistics groups organisms by common ancestry. Monophyletic is one. Para doesn't include all the descendants, and the poly distantly related does not include the most recent common ancestry. I don't know that I'm really going to get into this real specific. I'm not going to emphasize, let's just say that. I'm not saying I'm not going to ask anything related to it, but I'm not going to emphasize it. Shared ancestral character is a character that originated in an ancestor of a taxon. Shared derived character. This is where I wanted to kind of get to. Do phylogeny cladistics. This is, here is a set of characteristics. And here is, well, it's a listing of some different taxa. This is a lancelet. It is a chordate, but does not have a backbone. And this is a lamprey, which has a circular mouth. It's an external parasite, so it has no jaws. Here's a regular fish, here's an amphibian, here's a reptile, and here's a mammal. Okay, so if you ask the question vertebral column, how many of these groups have vertebral column? Right, 5 out of 6. So you put a 1 below each one that has it. How about hinged jaws? Lamprey does not have a jaw, lancet does not have a jaw, so 0, 0. Then you put 1s for these. How about having 4 limbs? Okay, well the bass doesn't really have truly 4 limbs. It has fins, but frog, turtle, leopard, they do, so 1's there. And development of the amniotic egg, which also an amnion is part of the placenta as well. In mammals, the turtle, which is a reptile, which is a characteristic of reptiles, is an amniotic egg, and then also a mammal. And a body mostly covered with hair, only the mammal. Okay, now if you look up and down, 1, 2, 3, 4, 5, you would say, you could add these up and say, the leopard has 5 of these characteristics. The turtle has 4. The frog has 3. The bass have 2. And the lamprey has 1. So let's ask this question. Which one is the most closely related to the leopard? Which one? The turtle, because it has 4. And then the frog would be next, the bass would be next, the lamprey, lancet's around. So that's kind of how that works. We need to quit. So, sorry to get further in this. Continue to read and study these. Be on the lookout for additional outlines that I'm going to be sending this weekend. 1 week from Monday is your first test.
Do we have another quiz?
We have a quiz next Friday.